Jeez, I don't know. Well, I got Peter up here first, anyway, it doesn't. Either way, we're here together. All right, we're going to kick this off. We're all ready to go. Thumbs up.
Wondering about the lighting. What do people think about lights?
Mood lighting,
Turn
or
off.
just for the sake of science?
Turn off the front right. Off the front lights if you can.
Yeah. All right, while we're getting our lighting adjusted, welcome again to the ninth, this is the ninth HDR Student Symposium for the same site. It gives me great pleasure to introduce our sort of esteemed leader here, Professor Peter McCluskey, to open proceedings.
So hello, everyone. Welcome and thanks for coming. I'd like to thank our panelists who got the hard part to do this afternoon. I think I could say that we all look forward to this afternoon because it's a really good opportunity to sort of see how broad our research programs are at the Institute and, you know, just the exciting cutting edge research that that's being done in the Institute. It's always a good fun afternoon. It's a good chance for us all to sort of catch up with each other and network. And I think Frank's been our MC for quite a few years now, always does a good job. So I look forward to hearing all the presentations and talking to everyone at our break and afterwards. So thanks again and welcome, and we'll get started. Over to you, Frank.
Thanks, Peter. Let's do this. Okay, just some housekeeping. Let me think. Bathroom, restroom. The female toilets are right behind us. Boys, you got to go for a bit of an extra walk around the block and you'll find the men's around there. If in the case of an emergency, follow the little green men all over the building. And I'm sure there's other instructions down the lifts or not. What else we got? Presenters today, 5 minutes each. It's going to be very nice and tight. There's a bell somewhere. Is there a bell? What's it? That'll be a 1 minute warning with that bell. So after 4 minutes, that you're going to hear with that little bell. It's going to be interesting. Don't let it put you off. Just stick to your plan. It'll be a little ding. Anyway, so I'll probably stand up close to 5 minutes and we'll get you to finish off. There's going to be a couple of minutes for discussion and questions. Welcome, Mark. And then we'll move on to the next speaker. So we're going to try and stick to schedule because that's how we run ship here. I think there is a slide that I might have access to or not. How do I progress? Oh, okay. We forgot. Well,
Just about the lectern and the clicker so everyone knows now.
okay. Well, obviously we're going to acknowledge country here. What are we doing?
Oh no, the slide is over there.
Which slide?
I mean, your slide is.
Yeah. Oh, okay. Thank you. I was getting there. All right. So opening remarks. We've done that. Tick, tick. That's me. Here we go. This is the slide I was looking for. All right. So as I mentioned earlier, this is 9 years since we've had this symposium, and we've seen a lot of great talent come and go. And over those last 5 years, I can happily say that we've had about 43 HDR completions, which is a great sort of reflection of the SAFE site and its contribution to the academic pool there. Special congratulations to these 9 individuals who, from our last HDR symposia, actually got their masters, or actually most of them a PhD conferred. So just a round of applause for a lot of those individuals from all different laboratories. So, without further ado, yes, I know, well ahead of time, but I'm not going to drag it out for 5 minutes. So are we allowed to start? Can we kick off earlier?
Just explain about all the lectern while you've got everyone paying attention at the same time.
What about the lectern?
Make sure
It's
you
up
pull your
here.
microphone.
Okay, speak in the microphone like I am doing. I'm setting a good example here. Don't go roaming. Where's Elisa? Stay put. Don't go roaming around. There is a pointer here. You can adjust. You can use the little mouse here. Apparently that works or not. So, anyway, so yeah, very easy. If you haven't been up here already, make sure. Well, anyway, too late. So, all right. I don't know. This thing doesn't work for me, so I got no idea what they're going to do. Is this advancing by itself, or is it this thing? There we go. All
You
right.
want to use the mouse for the pointer, or you can use the pointer that's in the advancer.
So you can tell why I keep invited here because Emma does all the work. So I know. Well, all right. Well, it's all self-explanatory. Every speaker's listed here. It kicks off at 12 at 1:40, so I don't know if I should start or not.
The
Oh,
Q&A for the microphone.
Q&A for the microphone. Above you are lights. Yes, everyone's Big Brother's listening. Those little green lights apparently are microphones. Apparently it's all being recorded and it's going to hear every word you said. That's why we put the microphones near Peter and John, because they're going to be asking all the questions, obviously. And there's some in no man's land over there. Anyway, we're going to be sort of speaking nice and loud, and so all our speakers can hear those questions and they'll be recorded. Again, Peter's already mentioned, we've got our esteemed panel here who's going to be judging everyone. And at the end of today's proceedings, there will be an awards, as Peter mentioned. We're going to give out 3 prizes for the 3 of the more outstanding ones. I'm sure they're all going to be outstanding, but 3 of the more outstanding ones will get recognition today. And I don't envy this table here. This is always a very hard decision. All right, how are we going? You didn't tell me I had 5 minutes of material. I didn't prepare anything else. All right, well, to stick to schedule, are we going to wait further?
No, I think we start with
If the next person's ready to start, Sam.
First speaker's ready to go? All right, let's do this.
Had some technical problems along the way.
We're going to definitely have technical problems from here. All right. So our first speaker is Samuel Clistomer. Is that good enough? Clistorna, sorry. Got to get one. And today, Samuel's going to be talking about evaluation of chronic lesion tissue in relapsing, remitting MS patients in association with disease progression. Thanks. You have the floor.
Sam, before you start, can you see your slides on the laptop there?
Yeah.
Yeah.
Good.
Is that presenter mode? We were just wondering if that's the full screen. Or
It's
not
full
slide.
screen, it's not presenter mode.
Looking good.
Good?
Yeah.
Okay. Good afternoon. My name
1 second, Sam, sorry.
is Sam. Today I will be presenting my final chapter of my PhD, and we'll be looking at lesion expansion in multiple sclerosis. Multiple sclerosis, or MS, is a neurological condition that primarily affects women from a young age. It is characterized by lesions in the brain and spinal cords. Here you can see an example of an MS subject across 3 time points. The white parts, or hypointense signal in the flare image, are the MS lesions. And we can clearly see the expansion of these lesions across the 3 time points. In this presentation, I'll discuss how we measure lesion expansion and use this method in a cohort of patients, and then explore its links and association to disease progression. So first, how do we measure lesion expansion across time? In our previous work, we developed a novel pipeline that analyzed the expansion of chronic lesions. Briefly, the fully automated pipeline uses lesion masks from baseline and follow-up images as inputs. A lesion mask is a type of image that highlights specific areas of the brain affected by lesions, helping us to isolate and study these regions. The pipeline excludes new lesions indicated by the green arrow, and then assesses each chronic lesion for expansion and positional shift due to brain atrophy. This process isolates chronic lesion expansion, represented in the red mesh. This is the methodology we'll be using for the study. The cohort used for this study was from our MAD-MS dataset. It included 68 relapsing remitting patients that reached at least 5 years follow-up, 63% which were female, average age of 41. 17 of these subjects also reached 7-year follow-up. Patients underwent annual MRI analysis, acquiring T1 and FLAIR scans. The lesion expansion pipeline was applied across the cohort. We measured lesion expansion annually and summed the results over time. An example of the pipeline output for a subject is shown here. The top row illustrates percentage expansion compared to the baseline volume, and the bottom row shows the volume increase over time. Here is another example. There are 2 key observations we can see. 1, some patients show no expansion across the 60 months. And 2, those who do show expansion follow a linear pattern. Next, Looking at the results across all 68 subjects, we averaged the expansion, finding constant linear trends for both the increase in percentage and also the volume increase, the bottom graph. Extending the follow-up to 7 years on 17 subjects showed similar linear results. These results show us that lesion expansion is potentially a constant linear process. Now, to validate whether lesion expansion is linear, we generated a line of best fit, or the dashed line, and calculated the root mean squared error for each subject. This measures the difference between observed data points and the line of best fit, with lower root mean squared error indicating a better fit. In our analysis, the median root mean squared error across the subject was less than 5%, meaning a linear model accurately represents lesion expansion in most patients. As such, we can say expansion can be modeled as a linear process, and we can say that subjects can be represented by linear gradient. And this is what we did. We assigned a gradient of expansion for each subject. Plotting the gradients for each subject shows high rates of expansion in some subjects, while others show no expansion. We separate the subjects into two groups: progressors, the top 25 percentile, and stable, the bottom 25 percentile. The average expansion rates for progressors show a clear linear trend. Running statistical models, we found that age and disease duration were not significantly different between these two groups. However, sex was significantly different, and there were 3 times more males in the progressor group compared to the stable group. So while MS is a female-dominant disease, there are more males in the progressor group. Next, looking at baseline radiological measures between the two groups, stars represent significant difference. We observed significant difference in brain volume, as well as white matter, ventricle, subcortical, and plexus volumes. Longitudinally, we looked at atrophy rates between the two groups. And again, we see significant differences in total brain atrophy, and also other measures such as grey matter, white matter, ventricle, subcortical, and plexus volume. So, in summary, we observed that lesion expansion is a subject-specific and a constant and long-term process, and subjects that experience lesion expansion are also linked with radiological measures of disease progression, such as whole brain atrophy. Thank you.
I ask, what's the reason to use root mean What's the reason to use root mean square instead of something like variance? What's the advantage?
I think they probably both perform a similar measure to see how well a linear model fits the data points. We just decide to root mean squared error. I'm sure variance would have done something similar.
Yeah. Sam, presumably these people were on treatment?
Correct.
Yeah.
We found no difference in treatment between the two groups.
Sorry, I had to repeat the question. Presumably these patients were on treatment.
Yes.
Yeah.
And there was no difference in treatment between the two groups,
Okay.
stable or progressors, but it could be because there's a large variety of treatment in MS and having a small sample.
Yeah. Because there's what, 8 or 10 different drugs they can use.
Correct.
Okay. Yeah.
If they are on drugs.
Yeah.
Some are not.
Thanks.
So you found a linear sort of progression in volume. So there was a linear progression in volume. Maybe I missed it, but how did you control for initial lesion size? So if a small lesion progresses by 1 millimeter, then it should kind of scale to the cube if you're doing volumes. And so smaller lesions, for the same sort of millimeter progression is going to demonstrate sort of greater volume change.
So the top row here, both the top are percentage increase from baseline, and the bottom is volume. So they both showed a linear trend. If you just look at the percentage increase, for example, if you had 100 voxels and you went to 110, it would be a 10% increase. If you went from 1,000 to 1,100, 10% as well. So the top row adjusts for baseline volume, and the bottom row adjusts for just volume change. you're saying because it's symmetrical, that the volume.
So what I'm getting at is if you've got sort of something that's a small lesion and it increases by 1 millimeter compared to a large lesion that changes by 1 millimeter, say in overall diameter, or radius, the volume scales to the cube of the radius, right?
Correct. So this is.
So a smaller lesion at commencement for the same sort of linear change in radius is going to demonstrate a greater change in volume than a larger lesion at baseline.
And so you would see a different percentage increase. Yeah, we see a linear trend for those subjects, even in percentage, because a lot of lesions are periventricular, so they don't expand as a balloon. They only expand, say, a small section outside in the white matter. Maybe I'm not.
Are they similar in size at baseline?
Between subjects, no. Yes, so I don't have the actual numbers, but there's 60-something% females in the baseline group. But when we're looking at the expanders versus stable, I think there were 3 times more males in the progressor group versus the stable group. So the disease is more destructive for males than females. Hormones. Yeah, so it's, pregnancy has a large impact, positive impact for MS progression. Yeah.
All right, no more questions. Thanks, Ed. All right, our next speaker on the program is Elisa Baldacano, and she's going to be talking about amacrine cells in the ganglion cell layer of the human retina.
It's down a little bit. Okay. Okay, yeah, okay. Oh, okay. Good morning, everyone. My name is Alyssa, and my talk is on displaced amacrine cells in human retina. In the posterior eye cup, retinal neurons process visual information and transmit signals from photoreceptors to bipolar cells to ganglion cells, whose axons form the optic nerve and send signals to the brain. Horizontal cells and amacrine cells are interneurons which modulate these visual signals. Most amacrine cells are located in the internuclear layer, and a smaller displaced population are in the ganglion cell layer, known as displaced amacrine cells, and these are the focus of our study. Amacrine cells are the most diverse and least understood cell class in the retina, as the function and spatial distribution of most types remain unknown. Increasing our knowledge of amacrine cells has therapeutic implications in the electroretinogram, which reflects amacrine cell function and dysfunction, and optogenetic therapy involving cell type-specific targeting for restoring vision loss. Our aims, therefore, are to firstly determine the spatial distribution and proportion of displaced amacrine cells, or DACs, and secondly, to morphologically classify DACs. We use post-mortem human donor eyes obtained from the Lions New South Wales Eye Bank, and all donors were aged between 31 and 59 years old. So here, this is a dissected retina of the left eye, and I'm pointing to the optic disc and the macula, which contains the fovea. For our first aim, we used immunofluorescence to determine the spatial distribution of DACs. So this is a flat mount, and we used antibodies against RBPMS, which is a ganglion cell marker, and DAPI, which is a nuclear marker, to distinguish the ganglion cells from the amacrine cells, which I'm pointing to. We also use other amacrine cell markers, and I'll go into those in a moment. For our second aim, we used intracellular injections to analyze the morphology of DACs. So here, the labeled cell somas are injected with dye, and this lipophilic dye will travel along the membrane to reveal the entire dendritic tree. So if we have a look at our first aim results, this is a vibrotome section through the central retina, and here is where the fovea is. And the fovea is a region specialized for high visual acuity in primates. So the central retina as well has a high cell density. So we took 100 micrometer thin sections to achieve better antibody penetration. So in green, these are the ganglion cells, and magenta are the nuclei. If you have a look closely, you can see that the DAC nuclei on top of the ganglion cell layer. But if we zoom into this section, you can see that further away from the fovea, there's more DACs. And we saw this in our quantification analysis. So 0 is where the fovea is, and you can see towards the periphery in the temporal and the nasal retina, the displaced amacrine cells outnumber the ganglion cells. So we quantified the entire displaced amacrine cell population, and now we want to look at specific types of DACs. So we immunolabeled the retina with antibodies against choline acetyltransferase, or CHAT, and that's an antigen expressed by the star-burst amacrine cells, well studied to be involved in direction selectivity. So out of our entire DAC population, we saw that around 31% of amacrine cells express CHAT. We also used antibodies against substance P, which is a neuroprotective neuropeptide, and secretogorgon, which is a calcium binding protein that facilitates insulin release. However, its role in the central nervous system is not well understood. And out of our entire DAC population, around 2% make up substance P, and secretogorgon is a little bit more at 6%. So if we have a look at our second aim results, we morphologically analyzed cells from 2 separate experiments. So in one experiment, the retina was labeled with secretogorgon, and in another experiment, the retina was labeled with substance P. And these are 2 different cells. So on the left, this is a micrograph of an injected cell, and on the right, this is a cell reconstruction. And in both experiments, we found that all cells had thick primary dendrites, long thorny processes, and stratify in the middle of the interplexiform layer. So we classified these as the same cell type. And we assumed that this cell would be expressing both secretogorgon and substance P. And we tested that by double labeling the retina with these antibodies, and we found co-localization of these markers in these cells. And we compared our findings to single cell RNA sequencing data in human, and we found a single amacrine cell cluster, GABA15, which expresses both the secretogorgon and the substance P genes. So from our findings so far, we can conclude that the proportion of DACs increases in the peripheral retina. CHAT cells or star-burst cells make up around 30% of DACs, and secretogorgon and substance P cells together make up around 7% of DAC, including the double-labeled cells. And a large proportion of displaced amacrine cells remains unidentified. I'd just like to thank my supervisors, Uli, Paul, Dario, and Ling for their ongoing support, and to Sammy and Felix for their help in their cell injections. Thank you for your attention.
Sorry, I thought it was a question.
Hi.
Sure.
From what you've done so far, do you get any clues on what the function might be of these ones that are displaced? Do they seem to have a different function or connectivity?
You mean
Then
displaced amacrine cells?
They're displaced amacrine cells as opposed to the sort of normal amacrine cell. Are they connect somewhere differently or?
Well, there's around over 30 types of displaced amacrine cells, but in general, like the inner nuclear layer amacrine cells are glycinergic, and they tend to have a smaller dendritic field, so they have a more localized response, whereas displaced amacrine cells, they're medium and wide field cells, and they tend to be GABAergic. They can also be found in the INL, but they have like a broader integrated response. But it also depends on the cell types, right? So the star-burst cells, direction selectivity, dopaminergic cells, light and dark adaptation, and A2 cells, scotopic pathway. So there's a lot of these cells, and we don't know the function of a lot of them. So, yeah.
Back to you.
Okay, very nice talk. Very impressive. I'm just wondering, because you have done the similar experiments across a panel of like different samples collected from different donors. Did you identify any like a big variation in terms of the expression and distribution of those cells in individuals?
Oh, between donors?
Between donors,
Yeah.
especially different age group, female, male. Have you seen any difference?
Yeah, as a whole, I didn't really see much of a difference because the donors that we tended to go for, so it was between a certain age group, no known history of eye disease, and the post-mortem delay was between 2 to 9 hours. So we tried to keep it controlled. And in terms of variation, I didn't really see much variation. I think it's quite similar across all donors. Thank you.
Mentioned that it's a large proportion of the DACs which are identified in your class. The transcriptome analysis that you did or you've had access to, does that give us any insights of what potentially makes up that large population?
Yeah, with the whole transcriptome analysis, yeah, there's been a lot of studies in human and mouse that have done single cell RNA sequencing, and now there's also spatial transcriptomics that show all the different amacrine cell types as well. But the only thing is it shows us what is there, but we're not sure exactly what these cells do. So I guess the next step would then be to find out what these cells are doing, and when we can find out what their function is, then we can find out what goes wrong in disease. So yeah, Did that answer your question?
Give me the identity of those other 60%.
Oh, the identity. Oh, there's so many.
So many, but you've had the different classes that you've identified. So
Uh-huh.
I'm just wondering, you got this unknown pool,
Uh-huh.
maybe there's.
Do you want me to list them? Yeah, A1 cells. Yeah.
There's probably
5
no
minutes
good
left.
protein markers as far as maybe for them.
Yeah, because they tend to overlap as well. So some genes are expressed in multiple
Yeah.
amacrine cells. So it's not just like one gene specific to one amacrine cell. So that's why that also makes it a bit difficult to study each individual ones as well.
Ballpark figure, how many groups do you think there are? Yes.
Of DAX. Well, in humans, it's found probably around 20 to 30, 25 to 30, yeah. Yeah, there's a lot, yeah.
No more questions? Thanks. Okay, moving right along. Our next speaker is Riva Nadkar. She comes. She's going to be talking about genomic editing approaches to resolve variants of uncertain significance to develop novel therapies for cone rod dystrophies.
Okay. So, good afternoon, everyone. My name is Riva, and I'm a second-year PhD student. And I'm going to be talking about my project, which is about genomic editing approaches to resolve variants of uncertain significance and to also develop novel therapies for cone rod dystrophies. So inherited retinal dystrophies, or IRDs, are the leading cause of blindness in the working age population in Australia and are known to affect around 1 in 1,000 individuals. IRDs are caused by a degenerating population of retinal cells, predominantly affecting photoreceptors and the retinal pigment epithelium, or RPE. And these conditions have a high genetic and clinical heterogeneity, with more than 270 disease genes known to cause an array of phenotypes, as summarized in the figure over here. And ABCA4 and POM1 are two key genes that are actually implicated in various types of cone rod dystrophies. And so for my PhD, I'm going to be working on these two genes, and the two aims of my project, in a very simplified way, involve reclassifying ABCA4 uncertain genetic variants, and then also designing and testing out genetic editing-based therapies for recurrent POM1 variants. And a major part of my project relies on modeling diseases, which I'm doing using induced human pluripotent stem cells, or iPSCs, differentiated into retinal tissues. So iPSCs are generated by first isolating peripheral blood mononuclear cells from a patient blood sample that are then reprogrammed into pluripotency and grown until confluent. This is the point where I differentiate iPSCs to encourage presumptive vesicle development and RPE development that starts occurring after about 4 weeks. I then dissect the vesicles and RPE, grow them until maturation, and then use them for characterization. And so RPE requires around 6 to 8 weeks to develop into a nice, dense monolayer of pigmented and hexagonal looking cells, whereas retinal organoids take around 30 weeks to develop. And this is one of the main reasons why I'm actually using RPE as a disease model for my first aim, because not only do they take less time to culture, but I'm hoping that they'll provide some great insights into what the function of ABCA4 might be in the RPE. And so I'll be doing this by first investigating known disease biomarkers in patient-derived iPSC RPE containing or carrying ABCA4 variants and comparing them to wild-type control individuals. And then I'll be examining the same biomarkers in RPE created from various types of ABCA4 variants, including uncertain variants and pathogenic and benign variants. And so I've introduced a whole series of these variants into wild-type control iPSCs using CRISPR-Cas9 homology-directed repair, which I first designed and then delivered to the cells using nucleofection. And so far, I've created 7 different lines for different types of variants, which includes 2 pathogenic, 2 benign, and 3 benign, and 2 ABCA for uncertain variants. And so this has been summarized in the table over here, and we were able to confirm that this nucleotide knock-in occurred based on a single peak shown in the Sanger sequencing. And so these lines are now at different points of the RPE differentiation process. And so one of the biomarkers that I first wanted to look at was mitochondrial dysfunction, which is used to measure active mitochondria present in both patient and control RPE. And so to make sure that what we're looking at is truly mitochondria, we use this mitochondrial membrane potential inhibitor to confirm this. And so, so we were able to see real mitochondrial staining, a clear reduction in mitochondrial staining in the patient when compared to the control, which kind of suggests that this disease model has a disrupted mitochondrial membrane potential. And this proves to be a really good biomarker, which I will next be applying to my CRISPR edited lines. For the second aim of my project, I'll be designing and testing various genetic editing therapy approaches for recurrent POM1 variants, as there are currently no therapies available for them. And so I'll be doing this in a 3-step approach, where I will first be introducing the variant into a cell line using CRISPR-Cas9 HDR, which I will then correct using prime editing and base editing. And I'll be translating this editing technique into the in vitro retinal organoid model for phenotypic assessment. And so in summary, I've been able to generate 7 CRISPR edited lines for various types of ABCA4 genetic variants. And I'm in the process of using various known biomarkers to investigate phenotypic changes. And so I've been using prime editing to correct this POM1 variant, and I've seen about a 40% correction activity. And so that's really promising. And so lastly, I'd like to acknowledge the entire iGenetics research unit, particularly my supervisors, and thank you all for listening.
Yeah, I'm not that mean. I didn't get up and kick you off. So, this paper's open for discussion. So these clones that you had earlier, you mentioned pathological benign. Is that something that you've sort of created, or is you screened them and said, oh, this is benign, this is pathological?
Microphone, please, microphone.
So benign and pathogenic variants and even uncertain variants are actually classifications determined by a committee or a group called the American College of Medical Genetics and Genomics and the Association for Molecular Pathology. And they have established these guidelines that categorize variants based on their functional aspects, based on a whole series of criteria points that actually determine if that variant is contributing to disease in some way or not. So based on those, based on what's reported in public databases, I've selected a couple of variants that that were interesting to me.
Very interesting story. But I'm just wondering, have you had any quantitative analysis to check the expression of all those knock-in genes, especially those variants? Have you compared the expression level to the wild type? And have you had any consideration how to normalize your readouts later for the molecular assays to assess the gene editing effect in terms of the expression level for all those variants?
Yes. So to answer the first part of that question, so there are different qualitative and functional assays that I'm currently working on. Particularly, I'm looking at ABCA4 protein expression changes across the various CRISPR edited lines in comparison to the control and also the patient. So those are some types of functional assays. I'm also looking at changes in mislocalization that could potentially be occurring from those ABCA4 variants. In terms of normalization, yes, there are so for example, with that mitochondrial assay, one of the ways that we're trying to normalize is by how many cells are actually seeded in that experiment or how many cells are present in that field of view based on DAPI counting, which were the blue dots. So there are different checks that we use for these types of assays. I hope that answers the question.
On the subject of that mitochondrial, is that just a mitochondrial marker, or is it indicating some sort of redox signaling which mitochondria involved with?
It's only a mitochondrial state. So, yeah. Yep.
Okay.
Thank you.
We're doing, Emma's going to be very happy. We're moving right along here, way ahead of time. Next speaker. Okay, so our next speaker is Azhar Arafat. And today we'll be talking about unraveling the molecular landscape of the retinal multiomics analysis of the human retina. Thank you, Azhar.
Hey, good afternoon, everyone. I'm Azhar from the Macular Research Group, and my talk today will be on unraveling the molecular landscape of the human retina through multiomics analysis. So as we all know, the retina is an incredibly complex structure. Its intricate design, from the densely packed cellular architecture to the complex molecular mechanisms, reflects just how fascinating this tissue is. Now, previous traditional methods have provided invaluable insights into retinal structure and function in both healthy and disease states, but they still fall short in fully capturing the molecular complexity. Now, thankfully, advances in sequencing and mass spectrometry-based technologies have paved the way for omics studies, yet these approaches alone, otherwise known as single omics studies, provide only a fragmented view of the whole picture. Now, interestingly, the bottom three emerging fields are now starting to reveal the deep molecular layers of the retina, but the number of studies remains limited, especially in humans, and most of them are single omics based, which kind of still fails to capture the interconnected nature of the retinal biology. Therefore, one of the comprehensive view is needed for this case, and one of the most the promising approaches to address this gap is through a multi-omics approach. So as the name suggests, multi-omics is integration of the multiple omics layers from different aspects of molecular biology. And the integration process itself would typically start off with a large set of multi-omics data. And these data sets alone will be really challenging to interpret without advanced tools. And so that is where AI algorithms come in. Now, these AI-driven techniques will then be able to learn the patterns of each omics layer to generate meaningful outputs, which later on can contribute to the development of personalized therapies for retinal diseases. And so because of this information, in this project, we wanted to obtain the multi-omics data sets of the macular and peripheral retina, analyze their interconnectedness, and integrate them, these multi-omics data sets using AI. So moving on to the next part, this diagram here shows our grand design of our research workflow. And to be more specific, we'll start by collecting 5mm punches of human retinal tissue samples, both from healthy and diseased eyes, focusing on critical regions like the macula and the mid-peripheral areas. We'll then perform untargeted proteomics, followed by targeted metabolomics and lipidomics using the LCMS. And the resulting data sets from here will then be processed using AI to uncover patterns and relationships across the omics layers. And it's worth mentioning that, well, the last one in particular, would be done in collaboration with our collaborators from the business analytics team of the UC Business School. That's it. Okay, so over the past year, we've been focusing on optimizing the mid-peripheral tissue collection and sample preparation, as well as conducting pilot studies to basically obtain the initial multi-omics data sets for testing purposes. Now, in a typical situation, 3 separate samples will be extracted for each of these omics layers, right? But we've developed a much more efficient approach, allowing us to extract all 3 analytes from a single small sample. And this is called the simultaneous metabolite protein lipid extraction method, otherwise known as simplex. So for each sample, the 3 layers were carefully prepared and run through separate LCMS to generate raw spectral data, which then will be matched through relevant databases using all of these specialized softwares and tools as shown in the diagram above. So for proteomics, we have identified at least 6,700 proteins through data independent analysis. For metabolomics, we identified approximately 205 metabolites from both helic and amide methods. And as for lipidomics, we identified at least 600 lipid species across 32 lipid group classes. And interestingly, the top abundance of each of these omics are linked to biological process and functions that are essential for maintaining homeostasis. We then performed molecular exploration followed by pathway enrichment and network analysis, which basically shows us and help us to visualize how these molecules correlate and interact with each other. So building on from our previous findings, the next step will be integrating this multi-omics data set. And of course, this will come with several challenges, including dealing with data heterogeneity and incomplete data sets. We're probably also going to need to refine our omics integration methods while ensuring that the biological models we develop are clear and interpretable. So where do we go from here? Well, moving forward, we focus on expanding our sample sizes and groups. We'll be selecting meaningful key features for dimensionality reduction, and then we'll integrate omics layers effectively and also train our AI models by consistently feeding them with new information to derive meaningful biological insights. So this slide marks the end of my talk, and thank you very much for listening.
So, question at the back.
Really nice talk, Azza. Where do you think, I mean, first of all, are you getting any phosphoproteomics data in your workflow? And also, where do you think this will fit in the landscape of spatial transcriptomics?
Right, that's a very tough question. Well, to be specific, in my project, we're only doing proteomics, metabolomics, and lipidomics. We're not doing anything with spatial transcriptomics yet. So what was your first question?
Like, are you able to look at phosphoproteomics? Yeah, like what's activated, what's phosphorylated?
Oh, so for now, we're not doing anything like how they activate or not, because basically we're doing untargeted proteomics, which basically just profile all the proteomes of the tissue. So for now, it's just kind of we're starting to establish a pipeline, which is great because it's working at the moment. And then the challenge from now is that how we're actually going to integrate all of these omics together. So phosphoproteomics, we're not dealing with that at the moment.
Thank you very much. Very interesting. So when you feed all your data into your neural networks, do you give the networks any a priori information, for example, about the structure of the differences between macular and peripheral retina and the cell composition and so on?
Yes. For the AI integrated the integration part, of course, we always have to feed them first with predefined studies at the moment. So the answer is yes, except that we're not actually at that point yet. So like to add more information on your question, of course, we do like more machine learnings and deep learnings, and also MOFA, which is multi-omics factor analysis integration. So yes, so all of the information we get from the macula, the periphery, everything all about the proteins, metabolites, lipids, maybe some imaging as well. So they're all integrated to that as the starting point.
Okay, thanks a lot.
You mentioned training the network. What is your idea of optimal output? What is the target for the training?
Right. So as I mentioned earlier, we're going to feed this with machine learnings, deep learnings, and other omics integrative analysis. So for right now, the main output we at least want to achieve is potential new biomarkers, pathway analysis, and possibly therapeutic targets.
Leave with that. Thank you.
Thank you.
That's how I beat the microphone there. This thing doesn't like it. All right, so our next speaker is Samaratha. And today I'll be looking at modeling the drive signal for a prosthesis in visual cortex. Just use the remote, yep, just advance that way. All right.
So this is a model of the upstream visual system of the primate. So look at the macaque monkey. All of the data in this talk are synthetic. So all the neurons are synthetic. And we're looking at the development of chromatic selectivity at primary visual cortex, or layer 4C beta. So we'll be looking at development, or this idea of development in the context of synaptic weighting. So how synapses are modulated in their strength over the course of development, from immature to a mature cortex. And development occurs in these two phases. The first phase being prior to the onset of vision, and the second phase being after the onset of vision. So the first phase is with these retinal waves that are intrinsically generated in the ganglion cell layer. And then after vision onset, it's with the natural scene, either when the eyes open or with birth. So images from the natural scene. So in this talk, I aim to construct this monocular signal processing model, which includes all of the cell types up until layer 4C beta, and has a plastic synapse between LGN, or lateral geniculate nucleus, and V1 neurons, or layer 4C beta neurons. And the modification of these synapses is via a Hebbian process. So it's a correlation-based process. And the ultimate goal here is to reproduce response properties of cortical neurons, both achromatic and chromatic response properties. So the model structure is as follows. The retinal cell types for the cones, bipolar cells, and ganglion cells, amidst others, but relevant to this talk, these are the ones. And this forms the off and on pathway at the bipolar cell level. Off and on pathway respond to opposite polarities. The response polarities are opposite, so one responds to the onset of a dark stimuli, and one responds to the onset of a light stimuli. There's a relay at the lateral geniculate nucleus, and then convergence into the cortex, where the off and on pathways cancel each other out due to cancellation. So we'll focus on this LGN to V1 synapse. So what is this Hebbian development process? Initially, we start off with an immature cortex where all of the synaptic weights are equal, and the inputs cancel out, which results in a very small cortical signal. So we need some process to develop the cortical response. So over time, we strengthened the on and off synapses based on whether they correlate with the cortical output. So in this case, the on, or the red, correlates with the cortical output and the off doesn't. So we strengthen the on synapse and weaken the off. And over time, we get a stronger on input, a weaker off input, and resultingly a stronger cortical output. And we represent this with the size of each dot. So a smaller dot means smaller synaptic strength, a bigger dot means a stronger synaptic strength. So if you remove the arrows and look at the entire input space, we get something like this. And this represents something you would see before any development has occurred. And if we take a circle, which represents the dendritic field radii of that cortical neuron, we can attenuate the weights based on a Gaussian distance-based attenuation. So if we look at the development of these input space over time, we show it a series of retinal waves, which you represent with drifting sinusoidal gratings, which are an entire stimulus set. And over time, so if we zoom into this, over time, these synapses will grow or reduce in their strength. So as development progresses from early to late in development, we get selective strengthening of off and on synapses. And we can see that there's an on and an off subfield which emerges. And from this, we get orientation selectivity, which is the first stage of feature selectivity in the visual processing pathway. So we can kind of characterize this a bit more and look at the orientation selectivity before vision onset. So as development progresses, the strength to an oriented stimuli or a graded, an oriented bar grading increases over time. So the response amplitudes to an orientation become much stronger, and this neuron is selective to a particular orientation in the visual field. So the second phase of development is with natural images now, so natural images from the scene. And the second phase introduces a chromatic signal, and this also captures more complex spatial features in the visual scene. So before visual onset, the chromatic response or the response to a chromatic contour is weak, and it increases after the second phase of development. So to summarize, the two-phase Hebbian development accounts for realistic response amplitudes in cortical cells, orientation selectivity, and also chromatic selectivity. Thank you.
The model predicts sort of the surface size of skin.
The size
Because
of
you would assume that sort of the a greater field with chromatic.
so
What was the question, please?
the question I think was
Does the model come up with sort of the spatial frequency of the
so each cortical neuron that we generate has a receptive field structure and property. Within that, it has a spatial frequency it's responsive to, as well as a defined size within the visual field. Does that answer the question?
Yeah, but is it different for achromatic and chromatic?
Yes, it is. So the chromatic response and the achromatic response are different, and it changes across the population of the cortical cells. So some will be more responsive to colored contours, some more to achromatic contours, and some equally.
And it matches what actually happens.
It matches in every way except one, which is the chromatic signal is weaker than expected in real cortex. So there is something that needs tweaking, I guess, in the model.
The chromatically specific cells, they're the ones, the cortical cells, are they?
Yes.
And how many are there? How many colors, how many specific colors are there that can be sensed by these cells? Like there's red, you showed red. How many other color sensitive cell types are there?
So this is the midget ganglion cell parvocellular system. So it's mainly red-green opponency. There's no real blue-yellow opponency, even though there are some S-cone connections into the surrounds. So we're looking at red-green opponency. It's hard to correlate that with the color percept per se. It's more of a functional distinction.
Yeah, another nice presentation. My question is there a difference between sort of the L&M sort of and the S-cone sort of, I mean, in the way it develops with the reinforcement through the pathway?
There's a difference in the connectivity and the density in the retinal arrays which we have. So there are more L&M cones and less S-cone. But in terms of the development, no. The
S-cones usually come first, don't they? The L&Ms arise later in the retinal development. Is there sort of any effect of that on the reinforcement?
We don't have that modeled in. We assume that they're all onset at the same time in terms of retinal development.
Thank you. Okay, this is the time. I have to leave it there. Thank you. Our next speaker is Xanthia Bell. And they'll be presenting on the visual field loss and driving initial findings from a scope.
Good afternoon, my name's Andy. I'll be talking about visual field loss and driving. Maintaining a driver's license is an important patient-reported outcome and contributes to an individual's independence and sense of autonomy. Inherited retinal disorders, namely retinitis pigmentosa and retrochiasmal visual field loss, most commonly from stroke, are two conditions which have the potential to affect licensing due to peripheral vision loss. There's currently no international visual standards for driving when considering complex and progressive conditions or adaptive strategies individuals may use, such as increased eye and head movements. Even in Australia, Ausroad has limited information, and the testing protocols may not adequately reflect or predict on-road driving performance. Misconceptions also exist in the medical and wider community regarding the appropriateness of people with RP to drive. And similarly, while some studies have found that there's an increased crash risk for people with a hemianopia or quadrantanopia, rates are in fact similar to those with other conditions such as cataract, which people are not automatically disqualified. As such, a scoping review is conducted to examine the nature and extent of the literature, which incorporates both visual field performance in individuals with an IRD or retrochiasmal visual field loss, and their driving status and or performance either on a simulator or on-road. The scoping review also aimed to examine the gaps in and limitations of the current literature. Multiple databases and other sources were searched, retrieving a total of 603 papers. After duplicates were removed, 478 studies were screened for relevance, of which 118 underwent full text review, and 16 studies were included in the final review. Looking at the 3 papers examining RP first, 2 were quite old from the 80s and 90s and found that accident rates were higher for individuals with RP compared to age-matched controls based on self-report and on interactive simulator. There was inconclusive evidence of the impact of visual field extent, however, with one study finding the horizontal visual field extent and binocular field area were predictive of car accidents, and the other study finding no correlation. In a recent paper published last year by the Lions Institute in Perth, retrospectively analysing data, 39% of RP patients met the fitness to drive standards in Australia based on acuity and a fixation monitored binocular estimate. Additionally, of the RP patients who reported to be driving at the time of data collection, only 52% actually did meet the standards. In this study, actual driving performance was not measured, so the effective visual field extent is not known. There are a proportionally greater number of studies examining the effect of retrochiasmal visual field loss on driving. 5 of these studies exclusively examined complete homonymous hemianopia, with no evidence of hemispatial neglect or other cognitive impairment. The main issue noted in these driving simulator studies was the significant blind side detection deficits that participants demonstrated, particularly as the eccentricity of the hazard increased. This was translated to potential increased crash risk at intersections and detection of pedestrians, which would be considered incompatible with driving. There was some evidence of adaptive behaviours reported, with one study finding people had compensatory head scanning patterns. However, 2 studies found there was no change to the scan magnitude. Another study found people took a lane position, which increased the safety margin on the blind side. The other 8 papers examined a wider range of field defects, including incomplete hemianopia and quadrantanopia. Overall, there was great variability in driving performance, and there was inconclusive evidence of the impact of visual field extent on driving. On a group level, often driving performance was worse to normally considered controls. However, on an individual level, many people were rated as fit to drive across multiple studies. Only 3 specifically commented on the effect of visual field extent, and the findings were contradictory. 2 found fit to drive individuals based on simulated performance had a larger remaining visual field area, and the other found that the type of visual field defect was not predictive of driving performance. 4 studies also commented on adaptive behaviours, noting increased head movements and longer saccades in fit to drive individuals. So in summary, there's inconclusive evidence regarding the impact of visual field extent on driving, particularly in the RP group. There's also conflicting evidence regarding compensatory or adaptive strategies people use. In addition, none of these studies consider the roving estimate, which can be used for licensing. So the next step is to perform a patient-based study to address these gaps in literature, to specifically examine the effect of visual field extent on driving performance. Participants will undergo visual field testing across multiple modalities and complete measures of cognition, which will be compared to performance on a driving simulator. This will help develop a model to form a toolkit of assessment devices that enable clinicians to more accurately assess whether their patients meet the standards for driving. There also remains the unanswered question of the minimum visual field extent on a fixation monitored estimate, which corresponds to a full roving estimate, despite the inclusion of the roving estimate in the Ausroad's guidelines, which will also be addressed in the study. Thank you.
So to find out if, for example, the current standard is like a 20-degree field which is 20 degrees up and down, or a band, which is 120 degrees wide, centered on the center, right? That's a fully specific thing. So you suggested some people might be safe to drive that didn't fulfill that criteria. Is that correct?
So currently the roving estimate can also be used. So even if people don't fulfill the 110, 120 degrees,
Yeah.
if they're able to look around during the test and pass based on the stance while looking around, they can still hold a license. There's actually no correlation between how the fixation monitor estimate corresponds to a full roving estimate.
But
if those people who pass.
You find out where some sort of cutoff whether people are safe or dangerous to drive, you have to explore around that boundary and allow people who weren't safe to drive and have an accident.
So the idea of using a simulator first.
Oh, let's see how
That's what happened. 52% of them.
So the idea of using testing on a simulator first to see if they are safe to drive.
You could then study one.
Different lighting conditions.
Yeah, so the driving simulator can be, they believe, can be put down to low light white driving as well and driving in that light.
Okay, thank you. So the last speaker for this half of the session is Miriam Raza, and she'll be looking at comparative electrophysiological findings in different retinopathies. Thank you.
Hello, everyone. As we all know that inherited retinal diseases are increasingly being associated with mutations in retinal development genes, it is crucial to understand how these genes interact with other transcription factors in this pathway to find out possible therapeutic measures. This study looks at retinal dystrophy associated with OTX2 gene. OTX2 is a developmental gene and a key regulator involved in the development of forebrain and eye. It is involved in the genesis of photoreceptors, bipolar cells, and RPE specification. OTX2, in combination with NRL and NR2E3, has roles in determining cone and rod photoreceptor fate. Downstream transcription factors also have roles in bipolar and RPE cell fate. Our clinical case is a 32-year-old female with a history of poor vision and high myopia since primary school. Genetic analysis revealed novel heterozygous mutation in OTX2 gene. Okay, a bit about NRL gene as it follows the same hierarchy as OTX2 gene in determining photoreceptor cell fate. NRL gene in association with NR2E3 is crucial for finalizing rod cell fate by inhibiting cone pathway. Autosomal recessive mutation in this gene leads to enhanced S-cone syndrome. Typical ERG findings consist of diminished rod response, broadened A wave, electronegative B to A ratio, pathognomonic response, and increased S-cone response. Coming back to our OTX2 associated retinopathy, retinal imaging of our OTX2 patient showed similar features as enhanced S-cone with clump pigmentary changes, loss of photoreceptor complex, and RPE atrophy. The full field ERG for our patient was available at 2 time points, 2013 and 2024. Initially, there is a broadened A wave and electronegative waveform. OTX2 has a role in specifying bipolar cell fate via VSX2 pathway or NRL pathway, which raises the possibility as to this mutation contributing to the reduced B wave. Follow-up full field ERG for our patient in 2024 showed evidence of progressive reduction in rod and cone responses with further reduction in A wave amplitude and electronegative B to A ratio. Whether the reduced B wave in OTX2 associated retinopathy is associated with NRL pathway or downstream VSX2 pathway is something to investigate in future studies. The early full field ERG of our patient also showed some waveform form shape similarity to enhanced S-cone syndrome, as you can see the third one there. To date, only 12 novel heterozygous OTX2 mutations leading to retinal phenotype have been reported, and our patient is the 13th one in this whole entire world. These results provide further insights into the genetic pathway of developing retina. Thank you.
The big one.
It is a big one. If you look at the first diagram there, so it is the main regulatory gene that is responsible for the activation of all the transcription factor downstream during the retinal development. So yes, it is the main one. So if when OTX2 is affected or mutated, then the RP atrophy is something you will see. There is reduced bipolar response. There is or you can also see some, definitely there is diminished rod response as well, because it is involved in the regulation of NRL, and NRL is important for rod cell death. So we see all this together. But if we compare the OTX retinopathy with enhanced S-cone syndrome, you wouldn't see exactly the same thing. You don't see the pathognomonic responses. So basically, pathognomonic responses are the one where you have the same waveform to LA3 and DA3. If you could see the arrows there, sorry, the arrows there. So this is the pathognomonic response that you don't see in OTX2. So this is our OTX2 patient, the top one and the second one. The top one is 2030 and the second one is 2024. So you can see that we do see the broadened A wave and electronegative waveform, but there is no pathognomonic response that you can see. And also the implicit time of A wave is also not affected. It's normal, so it's not delayed as compared. So it means that there is a normal phototransduction happening, but it's reduced. So that means the photoreceptor cells, it could be because of because OTX2 is involved in RPE maintenance. So if and it's highly expressed in RPE. So if there's no OTX2, that means that RPE function would be affected. And if there are no RPE cells to maintain the photoreceptor, that means that we will get the reduced photoreceptor response. So this is all it is about. Any questions?
Nice talk. Thank you very much. Based on the pathogenetic observation, all the things, is there any implication into the management or clinical treatment?
So at this stage, there is no treatment, but this is the main area. We are trying to understand this developmental pathway to find out that which part of this pathway we can target to find out the therapeutic measures, because there is, as I said, that there are only 12 cases reported in the published literature, and our one is the 13th one. So it's very rare condition. So once we have all this understanding, definitely this would be our next step to find gene therapy or any other therapeutic measure.
A slightly perhaps peripheral question, also a non-expert one. The enhanced S-cone syndrome. What's the current thinking in the field of why this signal is increased? I mean, obviously, there's probably not more S-cones, I can't imagine.
So what happens, I'll go back to my PowerPoint. That one there. So since OTS, enhanced S-cone syndrome, there is a mutation in NRL gene or NR2E3. So if you see that, and what happens that if there is no NRL, NR2E3 will have no rod responses. That's why you see the reduced rod responses there. But then what it does that the cells that are fated, the precursor cells that are fated to become rod photoreceptor cells, they change their fate to S-cone. As a result of this, we get more S-cone response. And at the same time, NRL and NR2E3, it inhibits the cone pathway, as a result of which we get reduced LM cone response, but then we have increased S-cone response as all the rod cells change their fate to the S-cone cells. So that's why we get that.
Okay, yep, thanks a lot.
Thank you. Is there anything else? Sorry.
We're going to have plenty of time to ask you more questions during the break. So we're going to take 10 minutes now, so everyone can stretch. And there's water over there. That'll do for now. And there's also some nice. I don't know if Emma's been baking or what. There's some cake over there if you want to indulge. We'll see you 10 minutes, lots of toilet break, whatever. See you here 10.
There are a number of studies that have been done in the last 2 years using machine learning with keratoconus. So there are quite a few, but the key aspects that mine differentiates itself and provides some novelty are on 2 different fronts. All of these studies utilize image data, so they take pentacam or similar data. Mine uses tabular data, so I translate any kind of image data and demographic data into a table and input that through the machine learning process, which enhances the applicability because you don't need a specific device to apply it to. And the other key difference and novelty I present is that all of the other studies utilize normal patients as one of their classes when they're classifying. So a lot of them are a binary classification of disease versus non-disease. Instead, I've taken confirmed KC patients and put them into 4 independent classes. So I've utilized retrospective data from 3 multinational centres, one in Australia, one in Saudi Arabia, and also one in Germany. Today I'm presenting on just the final chapter, which was looking at the Saudi Arabian data and the Australian data, just to manage the size. I put in 27 variables. Some of them are demographic, some of them are clinical. And we got our readings from a pentacam, but they could be taken from any number of different devices, which is one of the novelties of this project. I developed an unsupervised learning model, which is basically a variational autoencoder, uses a Gaussian mixture model and incorporates a multi-layer perceptron as one of its hidden layers. I put in the 27 variables in a tabular data format. They get compressed through dimensionality reduction. We go through this process that I won't bore you with because I know everyone starts glazing over as soon as I talk too much technicality. But the really nice thing is at the end of it, you can see there's a cluster diagram, and I'm about to show you why that's so important. So the top left figure is we got our clinicians to classify our keratoconus patients into 1, 2, 3, and 4 against amlodipine. And then my model classifies them almost identically, around about 86, 87% accuracy. So if you look at the bottom, the really nice thing about that is I'm longitudinally able to monitor my patient progression. So here I've got a 17-year-old and then a 30-year-old male. So there's a 13-year gap between his visits. In the red circle, you can see there's almost no differentiation between his first visit and his second. In contrast, when we look at this female patient who was 14 years old and then 9 months later came in for her second visit, we can see that she's progressed in the top right corner, closer towards the second classification, which would be a point at which the surgeon would start to look at CXL. So what I've done with that is I got some funding through Future Vision Foundation, which was really exciting. I've developed a web interface called Ophthalmology, and basically it allows clinicians to go in, input their test patients. They can go through and select which of these patients. So in this case, I've used Sally Smith, who is a made-up name, of course. With 1 minute. I've put in the 2 patients together, and then through magic, the surgeon's able to get an outprint to show them exactly where each of the visits were across all of the metrics, so they can look at them from a quantitative perspective. But the really exciting part is it prints out this document, shows the surgeon where the patient's actually sitting, and we can track them over time. So we're using the power of machine learning to assist clinicians to better monitor and manage their KC patients. So thank you very much to Future Vision Foundation and to my amazing supervisors, Jing, Jared, and Chris Lodge.
One burning question. And Nicole, tell us what's the highlight of your PhD?
The highlight, I think taking something that people don't really understand a lot of, and we hear the term artificial intelligence thrown around a lot, and I think the ability to show how it can actually work to support a clinical decision rather than try to overtake it is a really important thing, and to overcome some of the, the clinical challenges with an enigmatic disease.
Great.
And also to submit it. That was a nice moment.
All right. Well, thanks for that. Unless you want to ask a burning question, I can say yes. All right, just go.
Yeah. You mentioned that it's invariant to the type of image that have the image taken. Would it be better? Would the classification be better if you just focused on one type of thing, like pentacam?
No, not at all, because I'm looking at topographic or tomographic. What was really interesting, there's obviously many other chapters to the thesis, but I kind of was able to prove through a random forest feature selection model that really a lot of the demographic features we look at, like eye rubbing, which is controversial, age, gender, etc., don't really come into play when it comes to classification, but rather really the topographic indices are the most important. Yeah.
We gotta go. You can talk later. Don't know each other. All right, our next speaker is Adrian Hunt, and he's going to be talking about outliers of treatment frequency in retinal vein occlusion.
Thanks. So, yeah, what I'll be discussing, my name is Adrian Hunt. I'm a retinal specialist and I'm more clinically based than laboratory based with my research. I use FRB data to see the real world outcomes of all the injections that you hear about being given for retinal conditions. So in this analysis, we looked at outliers of treatment intensity in retinal vein occlusion. It was a 24-month comparative analysis of practitioners. So just as an introduction, there has been some work done on how many injections you give and getting better visual outcomes with those injections, but it tends to be a bit confounded by disease severity. So the patients that receive few injections often would have, less severe disease. The patients that receive lots of injections have severe disease, and with that comes a large potential for visual improvement. So you've got that classic confounding thing. What are you really measuring when you're measuring how many injections and how much vision improves? So we had a multi-center international observational study using FRB data. It was 2-year outcomes, and we benchmarked the practitioners rather than the individual eyes. They had to have an established history of treatment. We had 29 doctors, 12 countries, over 1,000 eyes with retinal vein occlusion. The primary outcome was a change in visual acuity at 24 months. We also looked at mean change in central subfield thickness, that's macular thickness, time in range, so how long did patients have good vision during those 2 years? So I might have timings on this. And the final injection interval. So how did we benchmark the doctors? We used a funnel plot. So on the y-axis, you have the mean interval between injections. On average, patients were receiving injections around 2 months, every 2 months, whereas we found particularly relaxed outliers were giving them much less frequently. And we found 3 very intensive doctors that were giving them very frequently throughout the 2-year period. So at baseline, there are 2 types of RVO, BRVO and CRVO. At baseline, the groups were broadly similar in the intensive, typical and relaxed groups, except for the vision in BRVO, but we did adjust for those baseline differences. And we also adjusted for which VEGF inhibitor practitioners might have a preference for or regional availability. So 24-month outcomes, looking at BRVO first. The intensive group had 16 letter gains, going down to the relaxed group of 9 letter gains. In CRVO, it went from 14 down to 12, but then all the way down to only 5 letter gains with the most relaxed treatment. There was a significant difference in the adjusted macular thickness, the time in range as well. So more patients spent, or more time was spent by patients over the 2-year period if they were under an intensive regime with good vision. And what did it take to achieve that? Well, the last treatment interval, this is the last interval at the end of the 2-year study. The intensive practitioners were treating every 6 weeks still, which most of us in the retinal community would see as fairly frequent, whereas the relaxed practitioners had gone out to 10 weeks in BRVO or 12 weeks in CRVO. These were the models predicting visual acuity along the top. We've got BRVO, not a great deal of difference in the vision, but basically it does sit in that way you'd expect. Red is intensive, blue is typical, and green is more relaxed. In CRVO, you can see how the green really dips down in vision. So probably CRVO is most prone to undertreatment. So conclusions, at 2 years, the intensive practitioners were treating RVO using VEGF inhibitors with twice the frequency of the relaxed practitioners. However, their patients had gained twice to up to 3 times as many letters of visual acuity. And the outcomes in CRVO seem particularly vulnerable to relaxed treatment. Thank you. Any questions?
Very interesting talk, Adrian. So why do you think people have adopted the relaxed approach compared to the typical, I can understand sort of something intensive, but why relaxed approach if they're not getting as good a result?
Yeah, I think there'd be a few reasons. I think regional differences, availability, cost, etc. Then you'd also think, well, retinal vein occlusion is one of those conditions where you kind of reach an equilibrium, whereas something like AMD, if you don't treat it properly, it just gets worse and worse and worse. So some doctors might be more in the mindset that maybe 612 is okay. Others might be pushing hard to get to 6.6. So I think regional differences and doctors' sort of pursuit of perfection, I guess.
Is there, so if an effective vision, I guess, ways over edema or ischemia, is there any kind of evidence that there's been sort of some stepwise reason why the
Yeah, there wasn't really a difference in ischemia. Ischemia is pretty poorly recorded as well, because not many people do fluorescein angiography that much anymore.
It could be, I guess, you could compute it by that.
Yeah, converting to bad. Yeah, I think we haven't really found that so much that people, you can't really identify the moment when people tip over. You certainly do identify in the registry macular changes affecting vision. So getting pigmentary maculopathy certainly is detrimental to outcomes. But ischemia is kind of one of those ambiguous things where unless you've got a fluorescein, it's really only a baseline data point on the registry as well. So if people don't do a fluorescein day one, which often doesn't happen because there's lots of hemorrhages, so if they don't do that fluorescein day one and tick ischemia or non-ischemia, you don't have another opportunity to record ischemia. And then for us, you know, why did the vision get bad suddenly? Well, you'd just be speculating it was ischemic or not. Yeah. Questions? Thanks.
So our next speaker is here we go, Chris Ovens, and he'll be optimizing targeted therapies in optic nerve disease. Thank you.
All right, good afternoon, everyone. My name's Chris, and I'll be taking us through our study optimizing targeted therapies in optic nerve disease, or OTTR study. And I'll give my wife the kudos for the title there. She likes OTTRs. So, oh, there we go. So what are inherited optic neuropathies? Well, these are the three key ones that we'll be looking at in our study. So the first is dominant optic atrophy, which is the most common ION, typically a nuclear DNA disorder causing slow progressive visual decline. Optic dysdrusen, we think, is a dominant disorder with variable penetrance, leading to calcium deposition at the optic nerve head. And Leber hereditary optic neuropathy is a mitochondrial disorder leading to quite marked and profound visual dysfunction over months. Now, previously, this has been, this sort of area has been devoid of targeted genetic therapies, but that's starting to change. So we have two sort of competing antisense oligonucleotides in phase 3 studies for DOA. You can see them, the two groups down the bottom here. And also for Leber's, there's linatidine, Nalpavec, which is a Navarro-associated ND4 genetic therapy, which has also shown quite a lot of promise in phase 3 studies. So this then raises a few important questions, though, if we're starting to have these treatments available. And one of these questions is, well, how do we assess treatment response in clinical trials? And this is where we start thinking about biomarkers. So what is a biomarker? Well, it's objective, and it's an indicator of a pathogenic process or response to treatment. But to be used in a clinical trial as an endpoint, it needs to be measurable, it needs to be repeatable, sensitive to change over time, and it needs to make sense conceptually and operationally. And it's also got to be clinically meaningful for the patients and their lives. So what do we know about biomarkers in IONs? Well, not a lot, in summary. This was a meta-analysis that we just did in DOA patients looking at longitudinal data in any biomarker. And we've only got data on visual acuity, and it's only in 7 studies that we found. And the rate of change in terms of the pooled effect estimate was not significantly different to 0. So we don't have much data, and even the data that we do have shows that, you know, this is a disease that really doesn't progress much at all, which, when you're thinking about study design, becomes difficult, because then your control group is not really getting much worse over your study timeline. and the implications that that has for the treatments that you're trying to test as well. So the first part of our study is to look at natural history in these optic neuropathies through the lens of different biomarkers. Do some progress faster? Are some more able to detect change in a more sensitive or reproducible manner? And so this is just sort of a Cook's tour of some of them. So this is specular microscopy, looking at the endothelium and the cell morphology there. This is our Hiller vision, so volumetric representation of retinal sensitivity at different points. This is our photopic negative response, looking at specific ganglion cell function. And this is our flavoprotein autofluorescence, looking at the autofluorescence coming from oxidized flavoproteins in disorders of mitochondrial stress. And so then the second thing we've got to ask is, okay, well, what is the influence of genetic factors on a patient's natural history? And so this is data from one of the early studies, early cohorts from Cure in 1996, and you can see the quite marked variability of the different patients there. So if you took the sort of pooled result, you might get a trend line, something looking like that. But could there be different genetic subgroups here which underlie the quite marked clinical variation we see? And so the thought is that perhaps, missense mutations or dominant negative effects might lead to more rapid rates of progression, while perhaps nonsense mutations or haploinsufficiency mechanisms lead to quite stable clinical courses. So this is, just scratching the surface of what we need to understand about these inherited optic neuropathies, leading to future genetic therapies. But that's hopefully what we'll be trying to achieve over the next couple of years. Thank you.
Chris, nice talk. Why have you chosen an endothelial, like specular microscopy? Is there any published studies about that?
Yeah, so it's tenuous, but there was one study in Kern-Sayer, which showed that there were endothelial morphology changes due to the mitochondrial dysfunction. And I think the thought is that because it's so metabolically active, the endothelium, that you do start seeing sort of abnormalities earlier in these mitochondrial disorders. So that was the precedent for that.
Can you tell us a bit more about the flavoprotein autofluorescence?
Yeah, sure. So I can't tell you much more because I don't know much more yet, but this is one of the newer modalities that we have that is being used as an investigatory trial endpoint for some of these newer therapies that we're running as well. We've been lent one at Save Site, which is quite nice. But yeah, the thought is that with mitochondrial oxidative stress, you then get oxidation of the flavoproteins, and then they emit a higher autofluorescence signal. And then just like, your regular autofluorescence, you then get a, black and white or, heat map of that, and you can then quantify that and sort of segment it however you want.
And it's in the ganglion cell.
So it's not specific to ganglion cells, is my understanding. It's just a mass response from the retina. But I think the thought is if you've got specific, ganglion mitochondrial dysfunction, then yeah. But I think they're looking at it in sort of AMD as well, and a few other things. Yeah.
Thanks, Mr. So our next speaker is William Yates, and he'll be looking at inherited retinal disease, history, disease modeling, gene therapy.
Thanks. That's all right. Happy, Emma? All right. Good afternoon, everyone. My name is Will Yates. I'm in my third year of my PhD. I'm based at the Children's Medical Research Institute. I'll discuss my PhD topic, which Frank has introduced. So It's all right, I'll click. No, I can't click. Inherited retinal diseases are the leading cause of blindness in children of those and working age adults. The problem is there's significant clinical and molecular heterogeneity with more than 260 genes implicated in inherited retinal disease with variation in the phenotype and expressivity. We can see that the particular gene of interest is PROM1 that I'm looking at for my PhD, which is smack bang in the center, and it's associated with Stargardt phenotype, a rod cone dystrophy or retinitis pigmentosa, cone rod dystrophy, or in some cases in Leber's congenital amaurosis. And previously there were no treatments until the advent of adeno-associated vectors, where you can deliver a gene replacement therapy through this vehicle. So PROM1 encodes the protein prominent-1, and it's associated, it's important in the disc morphogenesis of the photoreceptors, so predominantly at the base of the rods and right throughout the outer segment of the cones. And it interacts with other important genes implicated in inherited retinal diseases, such as peripherin 2, EYS, and photocadherin 2021. There's also implications in the RPE and the autophagy mechanism of the retina and housekeeping. And it's associated with both autosomal dominant and autosomal recessive inheritance, of which that's the focus of my PhD. But interestingly, it is suitable for AAV therapy in the sense that it's 2.5 kilobases, well within the packaging machinery of the 4.5 kilobases that you need for an adeno-associated virus. So the aims of my PhD is to do a natural history of patients with autosomal recessive PROM1, but I'm going to focus predominantly on the scientific aspects of my PhD, which is disease modelling in both a mouse model and patient-derived retinal organoids, as well as develop a gene replacement therapy delivered by AAV. So we have these PROM1 RD19 mice, which they have a nonsense mutation on both alleles. And what we can do is we can do visual acuity, fundoscopy, OCT, and electrophysiology in vitro, and then ex vivo we can collect the eyes for histology, immunohistochemistry, protein analysis, transcriptomics. And we can see the RD19 or the Newton model is good in the sense that there's no evidence of PROM1 expression at the photoreceptor outer segments, and this is a cone outer segment marker. And at 4 months of age, you can see there's a significant reduction in rhodopsin staining, as well as conorestin, which is a cone marker, and significant thinning of the retina, so making it a very good model. The other model that we're looking at is patient-derived retinal organoids, where what we take is peripheral blood mononuclear cells, and they're reprogrammed by the stem cell facility into induced pluripotent stem cells, and then they're further differentiated by the team at the eye genetics unit at CMRI into retinal organoids. And what we can do is look at patients phenotypically and try and look at signatures that way. And this is the control patient where you have human PROM1 here, and our autosomal recessive patient here where you have a loss of PROM1. And this is the photoreceptor outer segment of these mature organoids. When we're looking at this in greater detail, these are fully mature organoids. You can see there's a significant reduction in the cone staining or green staining and a reduction in the rhodopsin staining. And we can quantify this over a number of different replicates. And you can see there's a significant reduction in our patient compared to our two controls in terms of the cone density per field, and also a slight reduction in rhodopsin. However, this doesn't reach statistical significance, making this a particularly useful model in predominantly a cone rod mediated disease. The last chapter of my PhD is developing a gene replacement therapy. So what we've developed is 3 different constructs of varying lengths with different promoters, and one being codon optimized. And we're co-injecting this with a GFP labeled, and we're injecting this in an AAV8. And what we're performing is a subretinal injection. We're then looking at these mice at 2 weeks post-injection, doing OCTs and then collecting the eyes. And we can see with the OCT image as well as the fundoscopy, we are expressing GFP, meaning that we are getting it in the subretinal space in the injected eye compared to the uninjected eye, and we're not causing any significant toxicity to the retina. When we've collected these eyes, we can see that in our untreated mutant, there's no evidence of human PROM1 expression to be expected. But in the injected mutant, we can see with this GFP co-labeling that we are seeing human PROM1 expression at the photoreceptor outer segments. And more so when we just have the injected mutant mice only, we are delivering human PROM1 where it needs to be, which is in the photoreceptor outer segments. So this is just some of the preliminary work of my PhD. And I'd just like to acknowledge the scientists in the laboratory and especially the postdocs and my PhD supervisor, Professor Jamison. I'm a clinician who's trying to learn science, and it's a difficult balance to be able to do both. But the team at the CMRI have been extremely generous with their time and patient with me. So thank you, and thanks for your attention.
Thanks for the questions. What are the specificity of the label there, or not necessarily the labeling, but with the injections, are you targeting the right cells?
Yeah, so that's what we've done with the AAV8 is we're trying to get this into the photoreceptors. So you can use an AAV2, which is what Lux-Turner, which is what's in commercial use, but that seems to probably get to the RPE, probably other cells as well. But you can, depending on the different AAV, there's everything through to AAV1 to 8, and then there's all ones that have been mutated, and you can actually specify which cell type you want to drive your AAV, your gene expression into.
And yours is on that point.
Looks like it.
You inject these subretinally?
Yeah, it's John taught me the technique. It's challenging. It's very challenging. Yeah.
Okay, well, leave it at that.
Thank you.
Very well. Moving right along, we now have Sophia Zagara, and she's going to be looking at improving pediatric uveitis vision outcomes. Up there.
The last slide. Afternoon, everyone.
Microphone, please.
Afternoon, everyone. I've presented a few times before. So I'm looking at pediatric uveitic glaucoma and cataract in the advent of biologic therapy. So I've published my literature reviews and a number of background papers, and now sort of for the past 12 months, I've been focusing on my actual further results from my studies that I've been doing. And just to highlight again why it's so important is that this is the kind of disease that young people can present with or develop as a result of JIA uveitis, which can lead to a lifetime of blindness. So I've developed a number of REDCap databases. And so the first one was the child glaucoma outcomes. And then the second one is a CONCA database, which has a number of surveys attached to it. So within the child glaucoma outcomes, what I've been doing is collecting data on all the patients that have been coming to our multidisciplinary clinic at Westmead Children's Hospital, as well as some historical data that's been collected previously from pediatric uveitis patients. And I'm looking at the demographics, uveitis diagnostic fields, glaucoma diagnostic data, and then their repeatable sort of data every time they come to the clinic, and their surgical repeatable data. And so that's leading to a whole database of clinical outcomes and data in that respect. And that's historical data. And then the second phase is actually conducting surveys amongst all the patients who are now sitting in the clinic. So this is looking at, there's 5 studies that are both quality of life, and they've all been validated. So 3 of them are looking at quality of life questionnaires, and then 2 of them are looking at vision-related quality of life. So what's happening now is that the patients and their carers are sitting in the waiting room and filling out those surveys. And they do it by way of this, so that they are scanning a QR code, and then they can tap into it and they can come back at any point. And hidden in that database, I've put a record ideas, which then I can come back and then correlate the 2 databases together to correlate what they're saying about how they feel about it compared to what I'm getting with my results in the clinical data. So one of the papers that I'm in the middle of writing at the moment is looking at cataract surgery with IOL in patients in JIA. And it's a cohort of patients with the cataract in JIA-associated uveitis undergoing phacoemulsification with IOL implantation, as this is quite controversial in the past about whether you implant an IOL or not. In our
campus we implant IOLs. Then looking at the cataract surgery outcomes with the IOL implantation of these patients associated with the uveitis. And what we're doing is stratifying those three groups into the ones that have been treated with systemic or topical corticosteroids only. So they're a non-steroid sparing group. And then there's the anti-metabolite drugs, which are our traditional drugs like methotrexate or mycophenolate. And then a third group, which are now the biologic drugs. So the JAK inhibitors plus or minus anti-metabolites, because most people that are on a biologic have to remain on an anti-metabolite as well. And the main outcome from this study is looking at change in corrected distance visual acuity in three groups. And then included in all that is all the ocular, post-operative and systemic complications. So this is just a table a graph to help sort of show how the patients are all being dissected into those groups. And there's, in my study, there's 141 children in the database. 81 of those patients have JIA uveitis and 60 patients have non-JIA. So those patients all have other diagnoses of idiopathic AAU, intermediate uveitis, TINU, Blau's, BKH, and scleritis. And then going down, there's 157 eyes in our study, and 64 eyes of those have had cataract surgery. And I'm in the process of working out exactly how many have had in those three groups. And just correlating it back to the data, a number of studies have been done looking at cataracts in patients who have topical steroids, and there's a 0.04 increased risk or eye risk per year in over 4 years. In one of the papers that was done, in another paper, they had 32 eyes in 24 patients. So our study is much larger than the previous studies that have been done. So my next step is completing this paper, and then I'm looking at, in particular, JIA sort of glaucoma in that those patients, and then we'll be looking at the surveys of the quality of life data and correlating those between.
That's really interesting, Sphere. How easy has you found it collecting the patient-reported outcomes?
As in on the quality of life? Or?
Yeah.
Well, theirs is just, that's the validated study. So they're saying about how they feel. Is that what you mean? Or?
I'm just saying it's quite difficult to get them. I mean, I'd like to get them, but the patients aren't all that keen on doing them. So like what proportion of patients are you getting completed?
So that's just starting. So they sit in the waiting room. So we've actually got a captive audience.
Yeah.
I've already had 13 fill out the full survey. So they're sitting in the waiting room in our clinic for about an hour and a half to 3 hours. And so that's a really...
So you're hoping it's going to work well, but see how it goes. Yeah.
The one's waiting longer.
They wait until they're finished.
The good thing is they come back. So the next time in the clinic, if they don't finish it, the next time they're in the clinic and I go out and remind them and I go out to so I go to the waiting room and I do announce and give out the pamphlets.
Yeah.
So I guess I've got a captive audience. And then we've also got a clinical nurse who's sending it out to them as well and reminding them that way.
Yeah.
Have you got, what's your sort of duration of the years that the data's been collected? Can you look at, pre and post biologic data?
Yes, so about 2017 on is our biologic data. And then there's another period where it's more anti-metabolites. So that's about 2009 to 2017. I found a difference. And then prior to that was more, it looks as though it was more topical and oral steroid. And it's quite obvious the surgical numbers within those periods.
Okay, thanks, Finn. All right, last but not least, we have Rachel's right here, who's all been waiting patiently. And she's got a very long title, which I'm going to let her read. Emma's still playing around there, so I'll let that go.
Okay. Hi, everyone. I'm Rachel. My talk is going to be on the validation of two patient-reported outcome measure questionnaires, the CAT-QUEST 9 short form and the PRE-QUEST questionnaire in Australian cataract population. A little bit of background to start with. So the Australian Commission of Safety and Quality in Health have suggested that we incorporate these PROM questionnaires within the cataract service pathway. And they've suggested using either the CATQuest 9 short form or the PreQuest. Now, the CATQuest short form is a validated questionnaire, and this has been done in multiple studies across multiple languages across the world. But very little is known about the PreQuest questionnaire. And how you go about validating these PROM questionnaires is there is a predetermined criteria that you use to assess the validity, the reliability, and the responsiveness of the questionnaire to the intervention being measured. And now the gold standard of how you validate these questionnaires is using a model called the RASH analysis. In our study, we looked at the psychometric properties of those two questionnaires and compared them to each other using this gold standard RASH analysis model. So we conducted our study, a pilot study at Sydney and Sydney Eye Hospital in patients undergoing cataract surgery using the English translation of both of those questionnaires, which you can see up on the screen here. So the CATQuest on the left is a 9-item questionnaire, and the PreQuest contains 12 items, of which 7 of them actually overlap with the CATQuest. And they look at questions like if you have any difficulty with performing tasks of daily activity, for example, watching TV or reading. In our study, we administered the questionnaires day 0 pre-op, 1 month post-op, and then if they didn't return a post-op questionnaire, they were followed up with a phone call. We administer the questionnaires sequentially, so the CATQuest first for 3 months and then the PreQuest. In our study, the results show that the baseline demographics between the two groups were comparable. So these included things like age, gender, and whether or not they were having first or second eye cataract surgery. A third of our patients had pre-existing ocular comorbidities, so glaucoma, retinal issues. The post-op questionnaire completion rates differed quite significantly, and you can see in one particular category it was much lower, which I will touch upon. And there was a variability in the mean follow-up rates. In terms of validity and reliability, as you can see up on the table here, the middle column is the acceptable values of the predetermined criteria. The CATQuest in green shows that it conforms to all of the acceptable values and fits it, the RASH model, while the PreQuest does so for every single one of those except for unidimensionality. And I will touch up on that in our discussion. In terms of responsiveness of both questionnaires, they were both responsive to the intervention being measured, which is the cataract surgery, and there was improvement in visual function across all items in both questionnaires, with a significant effect size identified for both questionnaires, and there was no preoperative floor or ceiling effects in either questionnaires. And so what essentially all that means is that the CATQuest 9SF satisfied all the criteria of the RASH model, while the PreQuest did so for all except unidimensionality. And when a questionnaire demonstrates multi-dimensionality, it means that it potentially measures a secondary trait other than visual function, which was the primary intended outcome of the study. Now, in terms of our study, one of the notable limitations was the handling of post-op questionnaires, which led to poor completion rates. I have a couple of theories about that. So one of them is that there was quite a significant group of patients with existing ocular comorbidities, which get followed up in subspecialty clinics, which our study didn't really target. So we looked at patients undergoing, routine cataract operations and then post-op clinics. And then a secondary theory was that, particularly in regards to the PreQuest, there was a lack of awareness among staff. So a lot of people were aware of the CATQuest, but not necessarily the PreQuest. But our study is one of the largest Australian studies to date that looked at comparing the CATQuest versus the PreQuest in the Australian cataract population, as well as in both pre and post-op groups. So the takeaway from the study was that the CATQuest performed better in all the metrics assessed in Australian cataract population, that it is a valid and reliable and responsive PROM tool for measuring visual outcome of function. But we do need to look at strategies that will improve completion rates of these post-op questionnaires. And one of them is potentially using an electronic-based form of administering these questionnaires rather than paper-based. I would also like to thank my supervisors for helping me out with this study, our CNC joiner, and the Sydney Eye Hospital Foundation for funding the study. Thanks.
What was the completion rate?
So Cat Quest was about 2/3 and Pre-Quest was about a third.
Third.
Yep.
Request because of this completion rate. So that
Sorry, what was that?
how important is the number of items on one of those questionnaires, and can that explain the difference in completion rate?
The good thing about the rash analysis, it takes into consideration the number of items in a questionnaire, so it accounts for that. And in one of the metrics assessed in the rash analysis, it looks at the weighting of different, so it takes all of that into consideration. I guess the only difference is the number of items would add to patient questionnaire burden, but not necessarily affect the validity of the questionnaires.
Established that the more items, the lower the completion rates. That's just the way it is with those sorts of, with proms, that's just the way it is. That's why they're always trying to keep the number of items down.
That's true. In the Cat Quest and the Pre-Quest, the difference is only a 3-question difference.
It's not a big survey.
And it wasn't necessarily whether they completed the questionnaire completely, is whether or not they returned the actual questionnaire, and that was low. So they didn't even return the questionnaire. And I think that was one of the problems in the study, the pilot study design.
Presentation. I was going to ask, if you had to choose one, which one would you use to help with sort of the waiting list or preparing patients for cataract surgery? If you're going to triage as we go along, do you think this will add extra information to helping triage the cataract waiting list?
Well, that's a very good question. And it's one of the issues with incorporating these prom questionnaires routinely is that there isn't necessarily a guideline at the moment that helps triage cataract surgery. I guess that's kind of the next step that we're looking at, is whether or not we can come up with a guideline that helps us triage. And I think it will help, particularly some of these psychometric properties that the questionnaires have demonstrated. In terms of which one's better, well, one of them fits all the criteria and the other one does demonstrate that there's potentially a second trait being measured. But the Pre-Quest does include questions like whether or not your current vision affects your driving. And I think that's one of the important questions in terms of triaging cataract priority.
Thank you.
Rachel, thanks. You've got the 2 questionnaires, but are there any areas that are better worded or covered in one questionnaire versus another? That is, if you were to do a hybrid questionnaire, taking elements from both the Pre-Quest and the Cat Quest, do you have a comment on that? Is there scope for that?
There is, and I think that is very doable, particularly with this rash analysis model. I guess we just have to work on it.
Sorry, can I just make a comment? I'm really pleased that Rachel is looking at this, because whether we like it or not, patient outcome analyses are really going to be very important in indications for surgery. I've started instituting that in my private practice, and the thought would be that if they're referred for cataract, they already start filling out the questionnaire, right? Which is what you're hinting at, John. And by the time they get in and get booked for surgery, we've got to sort of work out, are they needy? Do they have symptoms? Because we make in our clinical notes, oh, I can't drive at night, glare, and can't do fine work. But this may be a metric that we're going to be asked to do. So in our recent accreditation, we're coming up now in the next week, when we get accredited, the criticism was that we did not fulfill one of the standards. And that standard is a consumer-driven standard, and the prom is part of that. So we say, yeah, look, we do like everybody else. We write it in the notes, and the commission that gives us now the cataract guidelines indicate that we have to do this. So it's going to be done. And what our next step is to do it electronically. So you're right. You know, if they can sit there and go online and they're prompt and they move through all the questions, and at the end they get a big bottle of champagne image, and you know, hurrah, you've got thing, or you've got 50 cents off for your cup of coffee at the Courtyard Cafe, which is what we thought we'd incentivize patients. We get a better uptake. But yeah, Mark is right. It's pretty woeful, and our patient population is quite multicultural as well at the eye hospital.
Thank you. Welcome, John, to the lectern to close some closing remarks.
Thanks very much, Frank. Look, it's been a great pleasure to have the 9th session. And really, I'd like to congratulate all the HDR students. Have been really wonderful presentations this afternoon. We've had some really great questions as well from all the guests, the supervisors here. So I'd just like to thank you all for the time and effort that you put into this. And I saw Mark was about to hand out the drinks a moment ago, but he's stepped back from his bar duties there. So it's really left for our hard-working panel here to tally up, get their abacus out and do the numbers. And while we'll go and enjoy a drink. So thanks very much. It was a wonderful afternoon.